Hello, everybody. Thank you so much for joining us for today's webinar, Agricultural Science Essentials Vernier Tools for Modern Ag Education. We are so excited to jump into today's topic. I am joined by two amazing presenters. But before we hop in, I do just want to guide you to the bottom of your Zoom window. You'll see what looks like a little chat bubble. I'm going to encourage you to click on that chat bubble. And if you look at it, it'll have a little to section on the bottom with a blue button, and it might say hosts and panelists right now. Please go ahead and set that to everyone. That makes sure that as you ask your questions today, it goes to everyone in the room. I say this for two important reasons. One, because other people are probably wondering the same things you're wondering. So if you have a question about something, please ask us. It builds community. It encourages other people to ask their questions. It helps everyone learn. In addition, as we answer your questions today, we don't want to look like we are talking to ourselves. So we hope that you will share your question with everyone so everyone knows what we're answering. In an effort to practice the skill, go ahead and let us know in the chat. Let's see, were you at FFA last week? I'd be interested to know. Okay, James wasn't. Oh, Kathleen wasn't. They were at NABT. We were also at NABT. Colleen was, yep, Colleen was at FFA. Who else? You can also say neither. Colby was not either. Fair, fair. Very cool. Well, we're 2 minutes past the hour, and I do just want to hop into some housekeeping items before I pass things over to our amazing presenters. Housekeeping item number one, you will receive a recording of today's webinar. It usually takes us about 3 to 5 business days to get that edited. So as soon as it's ready, it'll be up on our YouTube channel as well as our website, and you'll get an email with that link. In that email, you'll also get a link to the resources folder, which has today's presentation, a certificate of attendance, as well as some free experiments. I'm going to drop that link in the chat as soon as I'm done talking so that you all can follow along. There's a lot of clickable links in this presentation, so make sure you take advantage of that resource. And then the other piece of housekeeping that I have is, as I mentioned before, we want to answer your questions during today's webinar. So please make sure if you have a question, go ahead and drop it in the chat. We have the fabulous Colleen ready to answer your questions. So please make sure that you have that to section set to everyone so that everyone can see your chat. And aside from that, I think that's my spiel. Stay tuned for the end because we are doing a giveaway of a sensor for everyone who's still online at the end of the webinar. So I will see you soon and enjoy the presentation.
All right. I was going to ask another quick warm-up question, which is, if you want to practice your chat skills here, what are your top subject areas that you cover in your agricultural science class? And you can just drop your answers into the chat. Today, we're going to be focusing a bit on subjects like soil, water, plant science. But do you cover those? Do you cover something else? Let us know. Animals, plants, natural resources, food. Excellent, excellent. All right. Well, my name is Sarah, and I'm going to introduce you to our agenda. We're going to have some brief introductions, so you can get to know a little bit more about myself and Colleen. We will introduce you briefly to sensors and software for data collection. And then we'll talk specifically about sensors that you can use for different parts of plant science, sensors that we have for soil and water studies, sensors for topics like microclimates, as well as the effect of light. And then we'll talk a little bit about our agricultural science with Vernier book, and other ways to find our resources. And then we can have a general chat, but please remember that you can ask questions at any time. So if you have a question as we're going along, drop it into chat. We can stop and redirect. If we don't hear anything on chat, then we'll keep rolling. And then also if you think of things after the presentation, also please don't hesitate to reach out. All right. Well, I will introduce myself first. My name is Sarah, and I'm a biology staff scientist with Vernier. I was a college professor for about 15 years. My PhD is in zoology, and I went to Oregon State University. I'm representing today with my very dinged up Oregon State sippy cup here. I taught everything from freshman biology to endocrine classes, animal behavior, vertebrate zoology classes, and neuroscience. And I was a former researcher in animal behavior and physiology with kind of a concentration in behavioral endo and neuroscience. But that's me. I now work with Vernier, and I work with a lot of not just biology teachers, but also a lot of agricultural science teachers. So nice to meet you all if I haven't met you before. And then we have Colleen. I'll let her introduce herself.
Hello. I've been working at Vernier now for 12 years. My background is purely biology. I have a bachelor's from Truman State University, which was one of the more rural universities in Missouri. I have a master's in science and science education from Montana State, which has an amazing master's program. And before that, I actually was working on a PhD in genetics, but transitioned into education because that's where my true passion was. And really wanted to talk about the more global scale of biology and specifically environmental science. And I could not do that when I was researching fruit flies. Um, when I left grad school and got my alternative certification in Texas, I taught for 6 years in Houston, where I taught grade level biology, pre AP biology, and AP environmental science. I also did a lot of outreach activities of working with students and teachers in low income situations. So, hello.
All right. Well, we'll get rolling. I wanted to introduce the app that we have available for most of our experiments that use sensors for data collection. And that's an app called Graphical Analysis. The basic version of this app is free, and you can download it for 5 different platforms. Actually, I should say downloadable, I think now for 4 different platforms because Chromebooks are special. And so for something like a Chromebook, but even also available for Windows and Mac, you can use the app in the form of something called a progressive web app, where it actually runs in a browser window. So you don't even have to download it if you don't want to. And Chromebooks, that's the main option for you, is to just run it in basically in a page of Google by navigating to graphical-analysis.app. So I put a link down at the bottom that should take you, if you're in either the Edge browser or in the Chrome browser, it will take you straight to a running instance of the free app. You don't need a password. The links for downloading, if you want to do a downloaded version on a different device, I have a hot link there in the presentation slides that you can access later. And as I mentioned, it's available for a lot of different platforms. You can get it for Windows or Mac. It runs on Chromebook. And then it also runs on things like smartphones, so iOS devices, Android, and also, excuse me, also iPad. And what we're going to be talking about mostly today are sensors from our line of GoDirect sensors. Now, the app works with older sensors. Your campus may have the older style. And I say older style just because of the style that we've made for many, many years. We still make the corded sensors that we call LabQuest style sensors. The GoDirect sensors have some nice advantages over those in that you no longer need an interface to plug into a device. You can plug them in on USB, or you can use them over Bluetooth. And so there's some cost savings. And in many cases, the GoDirect sensors have additional channels, additional features that give you more value for the sensor. So we'll be focusing on GoDirect sensors as we talk about different sensors that can be used for different types of data. But remember that we do still have the corded sensors, and those could also be used. So the corded sensors work with the graphical analysis app that we just mentioned. And then also they would work if you are working on the LabQuest platform, which is our standalone data collector. These sensors work with LabQuest 2 as well as LabQuest 3. All right. So I will dive into different options that we have for, I thought we'd start with plant science and talk about some different topics. Again, feel free to jump in with questions. But as we start with plant science, the most popular topics that we usually hear from teachers are ways to cover topics like photosynthesis and cellular respiration, which are often really difficult for students to visualize. Also topics like water transport in plants and how water travels through xylem tissue and is lost to the environment. We have ways that you can extract chlorophyll and other pigments from plants and do analyses related to plant pigments. And we also have ways that you can measure light levels for grow spaces. And we will talk a little bit more about the light stuff later, ways that we measure light. But I thought we would start with talking about usually one of the maybe in some ways more complex topics to talk about, which is photosynthesis. We have sensors that can measure CO2 gas levels as well as O2 gas levels. So our carbon dioxide gas sensor is fantastic for measuring the products of cellular respiration. It can measure decreasing carbon dioxide during photosynthetic reactions. It's an environmental sensor that is placed in a chamber with the plant material. It can also look at things like yeast metabolism. So if you want to look at fermentation processes, you can do this with this sensor as well. You can also use it for soil respiration, and you can use it for animals as well as for environmental science studies. So it has channels for CO2 gas, and it is, I should say, it is a gas sensor. So it's not going to be useful for putting into a liquid. You can't do dissolved CO2 with this one. But it has channels as well for relative humidity as well as temperature. So it's a pretty robust sensor that is incredibly useful. I didn't even think to put in decomposition studies. So we've played around with it with compost and recorded values. I even one time put some rotten cheese in a chamber and looked at how much CO2 was being produced from moldy cheese that I found in the back of my refrigerator. So there's a lot of fun things you can do with this one. Similarly, we have a sensor that can measure oxygen gas. And this one is great for looking at enzymatic reactions that produce oxygen as a byproduct. Again, using it for photosynthesis, cellular respiration, animal respiration. This one also has an extra channel built in for temperature. And using these together, you can do a very simple photosynthesis experiment or cellular respiration where you measure the change in those atmospheric gases under light as well as under dark conditions. And we did a webinar not too long ago, a couple months ago, Colleen and I, on photosynthesis and different ways to measure photosynthesis. So our classic setup is you can find it in our agricultural science with Vernier book. And we use, typically we use spinach leaves from the grocery store, especially the type that come in a plastic clamshell because they're usually very crispy. And you can put those nice living leaves into a chamber, shine a plant light on it. And this beaker here is in the way, just in case your light is producing a lot of heat. You want to make sure you have a heat sink. If you're using a very cool light, such as an LED plant light that doesn't produce a lot of extra heat, then you don't necessarily need the heat sink, but the heat sink can really help keep your environmental chamber from getting a lot of condensation in it. But you could also use other leaves. I have used any number of leafy vegetables that come from the grocery store. I've used kale, chard, lettuce. I've had different folks call and ask if they could use one of those live lettuce heads where they were taking off individual leaves because the leaf itself completely covered the bottom of the chamber. The idea is you just don't want to stack the leaves too much because you want to expose every leaf surface to some light. But it's a great way for students to visualize the biochemical pathways that are happening, because otherwise they're kind of happening in their imagination. This way they're actually able to see when the light is on, oxygen increasing, CO2 decreasing. And then under dark conditions, of course, you only have cellular respiration happening. And you can see the opposite happen as CO2 builds up in the chamber, oxygen levels will decrease. But a nice kind of a simple prep. I really liked this one when I was teaching. It was great for my freshman biology students. And I've done this prep with younger students as well, and it's always really popular. Another one that I mentioned is being able to look at water transport in plants. And that's something that we have also in our agricultural science with Vernier book. And this one uses a gas pressure sensor. You can cut just about any plant with a slightly woody stem. It doesn't have to necessarily be woody as long as it's fairly strong plant material that doesn't get crushed. But you slide it into tubing. The tubing is filled with water. You just want to leave a bubble on the end with the gas pressure sensor. You make sure that you've got a good seal between the plant material and the tubing itself, and that the cut stem is bathed in water. But as water is lost from these leaves, it draws water up through the tube, and it actually creates a pressure drop behind it. And you can look at the rate of that pressure drop to tell you how quickly water is moving from the plant leaves. So you can try this with different treatments. For instance, your students can try shining some extra light on the leaves. They can blow some warm air across it or increase the humidity. They can try different experimental treatments and see how it affects the plants. My students always would get a little excited with the heater that they would use, and sometimes they would put it a little bit too close to the plant material. And in those cases, you could actually see when the plant was stressed enough to close the stomata and quit losing as much water. And it was really neat to actually see that happen. But this is a pretty neat prep that you can do with just a gas pressure sensor. And the tubing is all included with the gas pressure sensor. All the tools that you need is included with the pressure sensor. And you just use a standard ring stand and some clamps like you'd find in a laboratory. I mentioned that you can also make extracts of plant material. And for this, you would actually use a different app, but also free. And that app is called Spectral Analysis. And this is our spectrophotometer called the GoDirect SpectraVis Plus. This is a fantastic tool for analyzing plant pigments that you extract using something like isopropyl alcohol. You can make a chlorophyll extract and actually show your students the absorbance peaks of chlorophyll. You can also show them absorbance peaks for other types of plant pigments like anthocyanins, carotenoids, and just about anything that you can extract from a plant, put the liquid into a cuvette and put into this device. You can see what colors are absorbed and what colors pass through the liquid sample. You can also use this for various enzyme studies. We have those in a variety of books. I put a hot link in here for more information on our spectrometers because we have more than one, but this one is a fantastic product for high school as well as intro courses in biology and chemistry at the college level. It's quite a workhorse of a device. The wavelength range goes from the near UV all the way into the red and up to almost the infrared range. So this is an absorbance peak for an extract in the blue line is an extract of spinach. And you can see the peaks for chlorophyll A and chlorophyll B. These are absorbance peaks. So this is the light that's being most strongly absorbed from the liquid sample. And then this one in the middle, the kind of yellowish green line, that's actually an extract from cherry. Just crushed up cherries. You can look at just about any vegetable or fruit that you can think of. This is a great one to have your students do investigations of things they buy at the grocery store. I once taught a lab where our campus was right next to a large grocery store with a fantastic produce section. And we were doing a section on plants, and I had them go to the store, buy different plant material, and come back to lab with it. I had some things already picked out, and then they could bring anything else that they wanted. And they made their extracts right there in class. I did put in a hot link to 2 different investigations that use this method. One is on visible spectra of plant pigments. That's a more general lab looking at all the different plant pigments. The other lab is a really neat analysis of chlorophyll content in different types of olive oil. And this is a really fun one to do with students to get them to understand some of the issues with fresh olive oil that's produced locally or at least domestically versus olive oils that are imported, in which case sometimes they are adulterated with other oil. They can actually look at the chlorophyll content of their sample to see whether it's actually truly virgin olive oil and also get an indication of how fresh it is, because recent pressings of olive oil will have loads of chlorophyll in it, whereas oil that's been blended with other oils will have less chlorophyll, or if it's been aged for a while, if it's just been sitting around, the chlorophyll degrades. And in some cases, vendors will dye it green to make it look like it's got chlorophyll in it, but you can do this analysis and find out that it's not in fact chlorophyll. So I put a link in for anyone that is interested in either of those labs. They're a lot of fun to do with students. All right, I'm going to stop talking and let Colleen take over for a little while on talking about
Right.
sensors for water quality and soil.
So, for measuring water quality, there are a lot of variables to look at when evaluating aquatic ecosystem. And 2 of the main ones that we tend to look at are going to be the pH of a system and the conductivity or total dissolved solids of a system. So for pH, we have a great Go Direct pH or Go Direct flat pH. Either one of these would work great for use of measuring pH. In basically, you can measure it on in the field or back in the classroom. Um, and you can do things such as measure the pH of runoff, things in hydroponic or aquaponic systems. We can do watershed investigations. We can do rainwater investigations, um, all sorts of different things that you can look at. And Go Direct conductivity, kind of the same thing, is that it's going to be measuring conductivity or how many ions are present in the water. And so again, this isn't sometimes it's not necessarily a indicator of health, but it can be an indicator of characteristics of the area that you're in and how mineral rich the soil is, and how many of those ions leach out of the soil into the aquatic system. Um, and yeah, again, same thing. They can be done measuring in hydroponic or aquatic systems as well, because there's a lot of times people want to use more purified water that doesn't have as many ions in it to help prevent growth of certain organisms. And this would be a great way to be able to monitor that as well. Other things that are important is turbidity. So we have a new Go Direct turbidity that was released just about 4 months ago. Um, and this is going to allow you to measure how cloudy the water is, either in the environment or from circulating systems. Or a lot of times we can also look at water filtration projects as well. Um, it's also good for measuring the turbidity of water after, say, you do an experiment with rainwater runoff on agricultural land and look at the effect of different types of plants growing in there and how that can affect the soil being washed away or eroded versus not. Um, and the dissolved oxygen is probably your most well-known water quality sensor here. So it basically is going to be doing the same function of what Sarah was talking about with the oxygen and CO2 gas sensors, this one's just going to be measuring it in water. So you can look at the amount of photosynthesis and cellular respiration taking place. You can monitor specific lagoons to see if they're becoming oxygen depleted or if you're getting an algae bloom, how that effect is happening. The other great thing is it really it actually has an embedded temperature sensor in it. So you can really look closely at this relationship between temperature and how much oxygen water can hold on to. And easily in a classroom setting, kind of have students demonstrate that just by using basically a jug with some water and some ice. It's really simple to do. Oh yeah. So the other things we can look at it is the biological oxygen demand and primary productivity. We can kind of see how active water is, and it kind of helps students really think about the amount of energy being fixed within the first trophic level in plants before it moves on to the next in the animals and the consumers that are eating it. Like I said, it has multiple sensor channels. So not only does it do our dissolved oxygen concentration, but it also looks at percent saturation. We also have a temperature sensor on it, along with a pressure sensor. So dissolved oxygen is affected by temperature and pressure. And so it is automatically compensating for those two readings, which means you don't have to go through a calibration process or recalibrate it every time it actually you move it to a new location. And I see, say 10 feet. So the DO can be submerged, but like what Sarah said, as soon as you go like 1 inch below water, the Bluetooth activity ends up turning off on it. This is a sensor that is in line to have data logging available on it, and once we have it, it can go deeper. Um, I would say 10 feet is reaching it a little too far. We know it's rated for sure at 1 meter for an hour, but beyond that, we can't exactly say for sure, especially because of the USB connection on it. It also, if you're in a more saline, if you're measuring saltwater or anything, it does have a salinity correction channel because salinity levels can also affect the dissolved oxygen readings. Next. Soil. Okay, so there is quite a few things that you can measure in soil with sensors. We can investigate moisture, temperature, a little bit of soil chemistry, and my personal favorite is looking at the microbial activity in soil because there's some pretty cool stuff that you can see, especially when a lot of students, younger ones especially, are still learning that soil is alive. So some of the soil sensors you will want to use on site, like basically in the field when you're doing these measurements. The first one is going to be the Go Direct Soil Moisture. So the Go Direct Soil Moisture is a sensor that has to be buried into the soil, and it has to be basically inserted into the same plane all the way across because you want to make sure it gets an even moisture reading across the tines there. And what it's measuring is it's measuring the volumetric water content of the soil. So it's basically saying what percent of the gaps of the air pockets within the soil are being taken up by water in those gaps. And so the range of it, I believe, if I remember correctly, is 0 to 45%. Basically, most soil will not essentially, once you get over 25% and close to 30%, you're looking at a soppy, muddy mess. So hopefully you wouldn't necessarily have to have readings at that level, but it can go that high. We also have a Go Direct temperature probe, which is great for measuring the temperature of soil at different levels. It's a really great experiment to show students. You basically just mark the point on the soil temperature probe on the temperature probe at different heights, and then you can insert it to those different heights and measure the soil temperature as they go deeper. If you want to even go deeper than that, we have a wide range temperature probe that is actually much larger. The Go Direct one, I'd say the stainless steel tine is about this long, while the extra long, the wide range is like probably about over a foot. So it gets you a lot farther in depth.
We had Colleen and I were teaching a workshop outdoor with teachers here in Oregon, and we have a type of ant called thatcher ants that build a dome that can be about 1 meter across and like about a 1/2 meter high. And we had a group of teachers that were using we actually brought them a Go Direct wide range temperature so that they could mark it on the stainless steel barrel for depth. And they were taking measurements all through the ant mound, which was really cool.
My favorite is measuring soil respiration, cellular respiration of soil. So the Go Direct carbon dioxide gas sensor that Sarah was talking about earlier for photosynthesis is great for being able to do this. The key is you want to basically have a chamber. And so we've built chambers basically by cutting off the bottom or technically it's the top of a container and then cutting a hole in the bottom of it. And then we sell these grommets separately that fit the carbon dioxide sensor. And then you can position it over different areas of soil and in different kind of situations. So when I first did this, I was looking at freshly composted soil that was in our garden on the roof compared to soil in a bioswale, compared to soil underneath a tree that was dying because of root compaction, and got to see very dramatic different microbial rates, giving us an indicator of the health of that soil. And not surprisingly, in the soil that was the compacted soil, there wasn't much life there at all. Um, and it can take any period of time you want to measure this, but we've done it in as little as 5 minutes and get really good results. The key, though, is to make sure that you move away all the dust that's on top of the soil and actually get down to bare soil. The other thing to keep in mind is if there's any plant matter there, there is going to be probably some consumption of CO2 as well. So having that in mind. And the other two sensors that you can use with soil, you want to use more back into the classroom environment. And for these, you don't measure the soil directly, you're measuring it more indirectly by creating a soil slurry, where you basically are going to have a one-to-one mixture of your soil sample plus distilled water. And that's the key, is that you want it to be distilled water, because what you're going to do is soaking that soil in that water is going to pull all the ions out of the soil. And then you can actually measure the pH of that liquid or the conductivity of that liquid. And it will give you the readings of basically what that soil level is at. For pH, I definitely recommend the Tris compatible flat pH over the regular pH for this, just because it has a flat junction versus an exposed glass bulb. So the chances of breaking it on like a small rock or a piece of detritus, woody debris that's in the soil is much lower. Um, and the Go Direct conductivity, it gives you a good idea of kind of soil, like basically soil salinity levels, of looking at what's happening to the levels of soil, especially if there's been a lot of fertilization going on it or a lot of excess irrigation in those lands as well. That's going to leave behind a lot of minerals, and you can basically compare different areas of land to that. All of these things I went into a lot more detail at a previous webinar we did with the dirt on soil science that Sarah so nicely linked in here. Um, got a lot more details on all of them, so I highly recommend checking those out.
Got one more for Colleen to talk about, and then I'll take over for light.
So we have this awesome little weather sensor that is fantastic for measuring essentially microclimates or any weather conditions in small areas that you want to have kind of a relationship, looking at the land around along with the climate or weather that is happening at that moment. Um, you can buy the sensor by itself, or you can also buy it as a system where it has this lovely little wind vane basically on it that helps it measure wind direction with it. We don't include a tripod because a lot of people are very specific about the tripod they use. And you can get any type of tripod that has a standard quarter 20 mount for like a picture camera or things like that. Um, and it's one of our sensors that has the most, I would say in a way, bang for your buck with the amount of sensors that are included on this thing. Um, we have the impeller to measure wind speed. We have magnetic sensor on there that measures the direction. We have a temperature sensor on there. We have a humidity sensor on there, and we have a pressure sensor on there. And all of those combined are able to be used to give out readings such as wind chill, heat index. You can put in your elevation to get barometric pressure instead of station pressure. We can do absolute humidity instead of relative humidity. I feel like there's at least one more that I can't remember off the top of my head. But there are a lot of things that you can measure with this. And this sensor does have the ability to data log. So you can set it up and have it collect data over a longer period of time without it being actively connected to a device. And that's it.
All right. So lastly, I think this is our last big section. It's not a big section, but we'll talk a little bit about sensors that we have available if you want to look at the effects of light. For younger students, middle school, elementary, we have a light sensor called the GoDirect Light and Color, and it measures overall light levels in lux. And it has channels as well for students to be able to do some experiments, maybe looking at UV, as well as reflectance studies of red, green, blue. These are just in arbitrary values. So that's why we recommend this typically for younger students to understand how color is reflected from a surface. But this is a nice sensor if you are working in maybe you've got a class garden or something with younger students. This is a really nice way to measure light, and it is useful for a lot of other experiments that you might be doing with younger students. If you want to get into something a little bit more sophisticated for things like greenhouse light studies or measuring light in an agricultural space, I would recommend looking at something like the GoDirect PAR sensor to measure photosynthetically active radiation. And this one has a tuned band of visible light. Of course, the PAR range is just about everything in the visible light range that plants would be able to use for photosynthesis. And it uses this one has a sensor head that is from Apogee, which if you kind of know some other ag industry stuff, this is a company that makes really, really nice sensor head. So this is a more expensive sensor. You wouldn't want to get one for every student. But it's great if it's on a very long cable so that you could actually mount the head up at the top of a greenhouse as well as out in a corner if you wanted to look, be able to move a sensor around the lower part of a greenhouse to look for light quality areas that might be more shaded versus in the center. But this measures photosynthetic photon flux density, which is how many photons are hitting a square meter within a second. And it is really fantastic for greenhouse studies, for looking at your growing space. Sometimes you may have a chunk of land outside that your students are able to build a garden in, but maybe it gets shade for part of the day. And this is a sensor that's also going to be having data logging added. So you should be able to log over, for instance, like a 24-hour period to look at how light is changing through the day. I did some studies of my own yard because I'd put out some garden boxes that every time I looked out the window, they were in the sunlight. And I thought, these are great spaces for growing. And then I couldn't figure out why nothing was growing until I deployed one of these. And I connected it to a lab quest in order to be able to record data over an extended period of time, and discovered that that area of my yard was actually in shade a good portion of the day, in spite of when I happened to look out the window. So it was not, I had not picked the best growing space, but this is a nice sensor to use. And this is one of the free labs that we have in our assets folder for you that we're sharing with you. This is kind of the classic photosynthesis experiment that I mentioned earlier, using, in this case, just a CO2 gas sensor, but looking at how distance from a light source can affect photosynthetic rates. So you conduct the basic photosynthesis experiment at different distances from your light source, and then you go back with the PAR sensor and you measure the light quality at each of those distances. And we recommend doing this with something like a halogen lamp, so you really have a nice broad spectrum of light. Again, if you're going to use a light that puts out a lot of heat, make sure you're using it with a heat sink, either a water-filled beaker, or if you have something like a tissue culture flask, that works incredibly well for a heat sink as well. All right, to shift gears a little bit, I wanted to mention that we do have a book of experiments. These are probably most suitable for an intro agricultural science class, but it's got around 30, I think it's 31 or 32 experiments in it. And they cover a lot of the topics that we already talked about. It's got experiments for water quality, soil quality, photosynthesis, cellular respiration, looking at germinating peas. We've got a fermentation experiment in here. We also have some basic experiments about electricity and power systems, as well as renewable energy. So there's a little bit of everything in this book. And the way that our books work is, once a campus buys just a single copy, you still have the license for it. So it's possible that your campus may even already have a license for the book, and we can help you access it. So if you happen to create an account on our website, it takes about 30 seconds to create an online web account at vernier.com. And I put in a hot link as well that has a guide for how to create it, but it's pretty self-explanatory. If you use your school email address, it may already, our website is designed so that it will try to pull whatever resources are associated with the domain name and add it to your account automatically. Doesn't always work perfectly, especially if the district uses all the same domain. It won't work that way. But make sure that you tell us the name of your school when you register for the account, and we will go through and match your resources to your account. When you're inside your web account, there's a little tool that you can use to send us a message to look at what assets you may have, and then we can manually attach them to your web account. If you're at a college, then you'd have to tell us your department, because book titles are associated with your college department. But if you're at a high school or middle school, it's tied to basically your school campus. But you may have books that you did not know a colleague purchased, and we're happy to connect those for you. Any additional questions? We had a few comments and just a handful of questions along the way. Anything else? We'll see if anything pops in on the chat. I wanted to mention that we do have professional development options. Today, of course, is a professional development option in the form of a webinar. You can also find videos on product pages that are available from our YouTube channel. And then also you can request one-on-one training as well. Oh, let's see. What is the state of total P measurement? Phosphorus? Colleen, do you want to grab that one? I know you have worked more with. That one usually requires a spectrometer, correct?
Yeah, so total phosphorus or phosphate, we don't have anything for soil specifically worked out. We only have it for water. Soil nutrients is something I've I've briefly looked into. The problem with a lot of the colorimetric soil tests, and it's similar to the water quality ones. Oh goodness, sorry, very large tree just fell. Um, uh, it requires some pretty nasty chemicals that can be very difficult to dispose of. So we try to avoid recommending those just because we know the cost of disposal of those things properly can be quite prohibitive. Um, but we do have a protocol for the phosphate test in our water quality with Vernier book. That's also something I'm happy to give you some information about. Basically, we recommend a test kit. I can't remember. It's either from Hawk or Lamont. I want to say it's from Hawk, and it's essentially the students will create a standard curve and with a known phosphate amount, and then yeah, exactly. Hot acid washes. One of the reasons I'm like I don't recommend doing um I think it's O phosphates versus total phosphates. The total phosphates a lot easier versus having to do acid wash. Done that, don't want to do it again. um uh but you would need either a colorimeter or ideally a spectrophotometer to do that experiment. I wish there was a sensor to make it easier, but there isn't.
I know that there are NPK sensors for soil out there in the marketplace. And that is one thing that every once in a while we get a request for those. So if it's something you feel strongly about, let us know and we can put a +1 on when we consider products that we might look into trying to develop for the future.
Yeah, because there's a lot of things out there colorimetrically for hobbyists that are pretty easy, just and use a capsule. The other thing is that because they're so prevalent and cheaper, it doesn't there isn't much of a area for us to go into with it. So just because of how easily accessible they
All right. I think it's time for our giveaway.
Yes. So as Sarah and Colleen were chatting with you all, I entered everyone's names into a random name generator, and the winner of today's webinar is Becky G. So congratulations, Becky. I'm going to go ahead and email you. I just need to get some shipping information from you. So keep an eye out for an email from someone with a domain email at vernier.com. If you don't see it, check your spam. But congratulations and thank you all for joining us today. I also have a survey link in the chat and would love to get some feedback from you all. So
All right. Thank you, Dylan, and congratulations, Becky. All right, don't forget that you can still get a hold of us if you think of things later. So Colleen and I are 100% of your life sciences department at Vernier. It's basically the two of us. If you call and you ask, you will get, most likely you will get one of the two of us. We do have colleagues that work with ag mechanics and engineering. So if you need ag mechanics or engineering, they will probably connect you with some of our engineering education folks. And of course, we have also folks for chemistry. We have folks that work in physics. But we are your life scientists related to all things environmental, biological, agricultural. So don't forget that you can reach us through the website. You can call us, you can chat. We have a live chat feature that's on anytime the company is open. So normal business hours. And then you can also catch us if you want to get an email just to me or Colleen. Biology@vernier.com is a great way to reach either of us because it's going to come to both of us and then one of us will grab it. So yeah, don't hesitate to get in touch if you have questions.
Absolutely. All right, everyone. Have a great rest of your Thursday. Thank you for joining us, and we hope to see you at another webinar very soon. So, bye all.
Bye. Thank you.
Bye-bye.